The overall goal of the following experiment is to identify genomic targets of KDM5A, Jared1A, RBB2 histone demethylase. This is achieved by preparation of cross-linked chromatin that is sonicated to produce DNA fragments of the appropriate size. As a second step, RBB2-bound DNA fragments are immunoprecipitated with RBB2 antibodies. Next, the recovered genomic DNA is amplified by PCR for sequencing. Finally, Illumina sequenced short reads are uniquely aligned to the human genome, and significant peaks are identified and annotated to the closest genes in order to identify RBB2 target genes. Results are obtained that show molecular functions associated with RBB2 target genes based on genome-wide identification of RBB2 enriched regions.
Hi, I'm Mike Bichardi from the laboratory of Dr. Elizabeth Benevolenskaya in the Department of Biochemistry and Molecular Genetics at the University of Illinois at Chicago.
And I'm Abul Islam from the Laboratory of Biomedical Genomics led by Dr. Nuria Lopez-Vigas at the Barcelona Biomedical Research Park, University Pompeu Fabra.
And I'm Danielle Duval.
And I'm William Richter. Both of us are also in the Benevolenskaya lab.
Today we're going to show you a procedure for chromatin immunoprecipitation and sequencing.
We use this procedure in our laboratory to study the binding site of histone demethylases.
So, let's get started.
To begin this procedure, grow approximately 10 to the 8th cells for each immunoprecipitation, or IP. Here, diffuse histiocytic lymphoma U937 cells are used and induced for monocytic differentiation with TPA for 96 hours. Following cell growth, add formaldehyde solution directly to the media to a final concentration of 1%. Then swirl the flasks briefly and allow them to sit at room temperature for 10 minutes. Then aspirate the media and rinse the cells with 15 milliliters of ice-cold PBS. Repeat this wash once. Next, add 6 milliliters of lysis buffer 1 to each of the flasks on ice. Then rock the flasks for 20 minutes at 4 degrees Celsius. Finally, harvest the cells using a cell scraper and transfer them to 15 milliliter conical tubes. At this point, the cells can be stored at -80 degrees Celsius. If the cells were frozen, thaw them out. Once thawed, spin the cells down and discard the supernatant. Then resuspend the cells in 6 milliliters of lysis buffer 2. Rock the tubes gently at room temperature for 10 minutes. Repeat centrifugation and resuspend the cells in 2.5 milliliters of lysis buffer 3. Next, prepare the suspension for sonication by placing it in a beaker of icy water with the microtip of a Branson 450 sonifier set to between 50 and 60% amplitude. Sonicate the solution for a 30-second constant burst and cool on ice for 1 minute. Repeat these pulses 10 to 15 times. Then, pipette the contents of the tube up and down and transfer them to a new tube. Continue to pulse and cool the solution an additional 5 times. Following sonication, add 10% Triton X100 to 1/10 of the solution volume. Then transfer the lysates to 1.5 milliliter centrifuge tubes and spin the debris out in a microcentrifuge. Prior to chromatin immunoprecipitation, or ChIP, save 50 microliters of the cell lysate as the input sample. Then, combine the cleared cell lysate with dynabeads pre-bound to antibody that have been previously prepared as described in the written protocol. Rock the mixture in addition to the 50 microliter input sample at 4 degrees Celsius overnight. Wash the beads with 1 milliliter of wash buffer. Then, use a magnetic stand to collect the beads and remove the supernatant. Repeat this wash 6 to 8 times. Perform a final wash with 1 milliliter of TE plus 50 millimolar sodium chloride. Spin the beads in a microcentrifuge. Following centrifugation, aspirate any residual TE buffer. Then, add 100 microliters of elution buffer to the samples. Immediately after, incubate the samples at 65 degrees Celsius for 10 to 15 minutes. Scratch the tubes against a 1.5 milliliter tube rack every 2 minutes to keep the beads in suspension. Collect the beads using centrifugation and the magnetic stand and transfer the supernatant to a PCR tube. Then, retrieve the previously saved input sample and add 3 volumes of the elution buffer. Finally, place the IP and input samples into the thermal cycler overnight at 65 degrees Celsius to reverse the cross-linkages. The following day, add 1 volume of TE buffer to the samples. Then, add RNase A to a final concentration of 0.2 micrograms per microliter. Incubate the samples in the thermal cycler for 1 to 2 hours at 37 degrees Celsius. After incubation, add proteinase K to a final concentration of 0.2 micrograms per microliter. Then, incubate samples in the thermal cycler at 55 degrees Celsius for 2 hours. Next, extract the samples once with 1 volume of phenol. Extract the samples a second time with 1 volume of phenylchloroform isoamyl alcohol. Finally, extract the samples once more with 1 volume of chloroform isoamyl alcohol. Then, add 30 micrograms of glycogen to each sample. Also, add sodium chloride to a final concentration of 0.2 molar and 2 volumes of ethanol to the samples. Incubate them for 30 minutes at -80 degrees Celsius. After incubation, spin the samples and decant the supernatant. Wash the pellets with 500 microliters of 75% ethanol, then dry the pellets and resuspend them in 40 microliters of water. To check the sizes of the DNA fragments produced by the sonication procedure, load 5 micrograms of the purified input sample in a 1.8% agarose gel and run the gel. The sonication procedure should produce fragments in the range of 150 to 350 base pairs. However, if the fragments do not fall in this range, the sonication procedure should be adjusted by varying the number of bursts and the amplitude. Prior to amplification of genomic DNA, check the size, concentration, and enrichment of the samples as described in the written protocol. To begin amplification of genomic DNA, obtain 34 microliters of the IP sample and 200 nanograms of the input sample. Perform end repair, A-tail addition, and ligation of adapters to DNA fragments using an Illumina genomic DNA sample prep kit as described in the written protocol. Purify the DNA using a mini-lute PCR purification kit and then elute it in buffer EB that has been pre-warmed to 50 degrees Celsius. Use 23 microliters and 46 microliters for the IP and input DNA samples respectively. Make the PCR reaction using 23 microliters of DNA with adapters, 25 microliters of fusion DNA polymerase from the kit, 1 microliter of 20 micromolar Sol PCR1, and 1 microliter of 20 micromolar Sol PCR2. Run the PCR reaction as described in the written protocol. Following PCR amplification, purify the DNA with the QIAGEN Minilute PCR purification kit. Pre-warm 15 microliters of buffer EB to 50 degrees Celsius and dilute the DNA. Dilute 0.5 microliters of the sample 1 to 4 and perform a nanodrop reading. To purify the amplified products, prepare a 1.8% agarose gel of 50 milliliters using HPLC grade water. Add TAE and ethidium bromide after the agarose has melted. Then pour the gel. Load the gel after adding 4 microliters of loading buffer to the input and IP samples. Then run the gel at 120 volts for 45 minutes. Analyze the gel after it has run. The gel should reveal that fragments in the range between 150 and 350 base pairs are produced. They represent genomic DNA fragments between 50 and 250 base pairs in length and an adapter. Excise the region of gel containing the material in the 150 to 350 base pair range with a scalpel. Take care to avoid excising the adapter band, which runs at about 120 base pairs. Recover the DNA using a QIAquick gel extraction kit per manufacturer's instructions. Dry the sample down to precisely 11 microliters in a speed vac without heat. Remove 1 microliter of the sample and measure the DNA concentration using a nanodrop spectrophotometer. Finally, identify enriched gene products using the Illumina Genome Analyzer and analyze the binding sites and target genes as described in the written protocol. Genomic coordinates of identified enriched RB binding protein 2 or RBP2 binding regions are presented in a chromosome-wise format. Occupancies of ensemble genes in the chromosomes can be visualized as black bars. Here, chromosome 10 occupancies are shown. RBP2 binding regions are shown as vertical lines. The middle panel shows all RBP2 isoform binding regions on chromosome 10, and the bottom panel shows RBP2 large isoform binding regions. This information gives an overview of overlapping and specific occupancy of RBP2 isoforms on chromosome 10. Here, a heat map is shown with false discovery rate, or FDR, corrected, significantly enriched gene ontology, or GO, molecular function categories among the genes bound by RBP2. Colors toward red indicate high statistical significance, yellow indicates low statistical significance, and gray indicates no statistical significance. Enrichment analysis shows the overlapping and isoform-specific molecular functions of RBP2 target genes.
We have just shown you how to identify transcription factor binding sites.
During this procedure, it's important to produce DNA fragments of the appropriate size range and yield. This can be achieved by following our guidelines during the sonication step.
So that's it. Good luck with your experiments, and thanks for watching.